Okay, so it looks like we've got the bulk of the people registered in the webinar. So thank you very much for joining me. I'm Jonathan Frampton, Director of Business Development at Proteonic, and the webinar today is navigating the viral vector manufacturing landscape. The format is it's about 35 minutes so of slides that I'll walk through. If you do have any questions, feel free to type into press the button on the questions and answers. Type in your question and I will endeavour to get to them towards the end of the webinar. There'll be a recording of this, hopefully, if technology works well, which I can share the link with you all once we've prepared that, so you can share with colleagues afterwards. So without further ado, so what I'd like to talk through on today for the webinar is the objective is very much looking at the challenges of viral vector manufacturing, and then looking at strategies that we can we can practically we can apply to optimize the increasing demands that is becoming apparent within the cell and gene therapy space, and then touch on technologies that could then support enhanced capacity, demand, and costs. The agenda, I'll start by looking at the current market landscape. What does it look like? Talk around the capacity crunch that is facing viral vector manufacturing. Then go through some of the costs that are associated with viral vector manufacture and strategies to reduce the costs of them. Then touch on the manufacturing process and then finish up with technological innovations. Now, before I sort of jump into the webinar, I thought it would be useful to give a little bit of background. That's just one slide on who Proteonic are. So we've been around 20 years now, and we're a technology company. We have developed vector technology for increasing transcription and translation of any gene in a mammalian system, which historically we've focused very much in the biologics space, so increasing expression of biologics in CHO cell lines. 2 years ago or so, we looked at how we can apply the technology that's been clinically validated into the cell and gene therapy space, and so we've now taken the proven technology and applied it to increase titers for lentivirus and AAV for the large pharma companies, as well as we're starting to work closely with CDMO companies, and through partnerships we are progressing and developing our technology very, very rapidly. And there's a nice sort of timeline of the different types of companies we have worked with. Okay, so back to the webinar and what we have at hand. So if you check, as of the end of April, there are 37 FDA-approved cell and gene therapies, and there's a broad spectrum, whether that's in oncology, solid or liquid, in neurologic, in cell transplantation, and it's only the breadth of cell and gene therapy is only growing. If you look at it from a monetary point of view, last year there was $10 billion spent on cell and gene therapy manufacturing. What is expected in 10 years' time is an increase of at least tenfold, with maybe $100 billion. Now, if you look at that or consider those numbers, for me, while it's clearly it means that there has to be at least 10 times more treatments or doses applied to patients, but in those 10 years there's going to be significant cost reductions as well. So capacity for treating patients could be anything from 20-fold, 30-fold, or even greater in that time frame. If you were looking, if you were considering that capacity fold increase for small molecules, it wouldn't be too much. It wouldn't be too much, or it would be like, okay, that's achievable. What's becoming apparent, and I'll show some data shortly, is the cell and gene therapy supply chain is very delicate, and even small increases of demand really puts pressure and limits. There's bottlenecks of being able to get treatments and therapies to the market. If you look at, if you dive down, so that was the macro look at the market. If you dive into, let's say, the myeloma treatment drugs, so you've got BMS and J&J with Abecma and Carvykti. In 2022, there was approximately 1,000 doses between the two of them administered. A year later, that had trebled to 3,000 doses, and by 2026, there was an expectation of around 22,000 doses being administered. So for this smaller area, you have to have an increase of 8-fold of capacity to be able to deliver the doses that are expected for the patients. Now, with the increase of capacity requirements, has actually driven what some people in the industry are calling the capacity crunch, where there simply isn't enough manufacturing capacity or capabilities worldwide to actually deliver cell and gene therapies to patients. So here are two examples. So you've got J&J's CARVYKTI's. They've actually had to pause. This is last year, March 2023. They actually paused the launch of the therapy into the UK market due to manufacturing shortfalls. And the cause of the manufacturing shortfall, or at least it was certainly attributed to the shortage of lentiviral vectors. Similarly, and this was a couple of years before that, BMS, when they launched Abecma, they had similar challenges. Once the uptake of the therapy started to increase, their supply chain was not robust enough to be able to deliver the actual the therapies to the patients. And again, the viral vector supply chain was attributed to those challenges. Now, there were some extenuating circumstances, because you did have the macro event of COVID and the pandemic, and a number of CDMOs being switching, or production being switched to the COVID vaccine. However, for me, it was more of a putting spotlight on how delicate the supply chain is versus a short-term issue caused by COVID. In 2 or 3 years' time, if capacity solutions are not addressed, then the next new cell and gene therapy coming through will have similar challenges. Because if you look back at a lot of the ones that have launched, any that have then become relatively mainstream therapies, they've all struggled with capacity and manufacturing issues. Up till now, what's been the solution? Well, whether it's Kite, J&J, Novo Nordisk, it's looking at, let's expand capacity through new manufacturing facilities or getting access to other people's manufacturing facilities. So Kite, for example, in 2022, they built out a whole new retroviral vector manufacturing facility. J&J have spent $500 million on a site in New Jersey, and similar money on a site in Sassenheim in the Netherlands, specifically for manufacturing their lentiviral vectors. They've also partnered with Legend Biotech, and in addition to that, actually signed up or sourced access to Novartis's supply chain as well. Catalent has been bought by Novo Nordisk for 16.5 billion. So Novo Nordisk are going to come into this of like, okay, they have a great manufacturing facility. Let's absorb that into Novo Nordisk to be able to deal with supply and demand in that fashion. So for me, it's everything is we are struggling with capacity. So let's replicate what we're doing and build out as best we can. Now, what does that actually impact? Or another way of looking at it is, it's clear to me that cell and gene therapy, the cost is not sustainable. I mean, these are sort of shock headlines that have come out, where you do have some gene therapies that are coming in at an acquisition cost of like 4 million. The way that they justify this is because there's only, I think, 20 patients worldwide that ever need this therapy, and so it is costly to administer. However, even when you get into CAR-T, and whether it's 500,000 or around the 500,000 mark, that is simply not sustainable for the wider population. There's lots of economic discussions around, well, if you're with CAR-T, it's a one shot, so you haven't got any of the aftercare. You don't have to go through first therapy, second therapy, tertiary therapies. So it is cost effective in that sense. But I think everyone can agree that if we can reduce the cost to enable these types of therapies for the mainstream, it will only be good for the patient. And over time, naturally, the costs will be driven down. If you look at the costs of cell and gene therapies, this is from Cytiva. This is a cost analysis of autologous CAR-T. you go look at the bottom of the slide, you've got in late phase clinical development. So once you've launched the product, got it to market, incredibly almost a quarter, so 23% of the cost is attributed to viral vector costs. You have some, you have almost 30% in labour, and then the rest is then made up really of media and reagents. But a quarter of it is in viral vector costs. And so if your plan is to build out new buildings to cope with the capacity, you are just going to be making that chunk of the you're going to make that chunk of the manufacturing costs bigger and bigger and bigger, and it's going to be an issue. I've realised my video is actually over some of the slides, so what I'll do is I'll just stop my video. You'll still be able to hear me. Okay. So looking at the viral vector workflow, it's just a simple schematic. You've got the upstream, the downstream, and the fill finish formulation. What I'm going to talk mostly about in this webinar is the upstream. I'll touch a little bit on downstream and the fill finish. And obviously, and within the upstream, you've got different types of cells you can use. There's the cell expansion, transfection reagents to increase transfection, and you then have the viral vector production. Because I'm coming from the proteonic angle, believe it or not, there'll be some focus, or quite a lot of focus, on the viral vector production as in these slides coming up. Now, one article that is actually it's a very interesting article that I recommend if you are into the economics of cell and gene therapy. It's written, came out of UCL, and they've looked at lentiviral economics. So really analysed what people are using right now in terms of bioreactors and different technologies, and what could be addressed to then try and reduce the costs in the future. The first question was looking at the choice of bioreactor, and as you can imagine, it is critical for long-term cost savings. If you are working with a therapy that requires a lot of lentiviral vectors, for example, a hematopoietic stem cell product, the contribution can be as much as 90% of the raw material cost. So the cost of the lentivirus is critical to how expensive that drug will be when it comes, that therapy will be when it comes to market. In their example, they were looking at, okay, if they could increase the scale by 200-fold, which would mean going from the small adherence, small 10-litre scale up to like a 2,000-litre scale, they could reduce the lentiviral costs from 9% down to 21%. So that could be a huge saving. There's obviously you are assuming that I mean, most people now are already working with suspension cell lines, so that's the switch from adherent to suspension shouldn't be too much of an issue. 3, 4, 5 years ago, that was more of a challenge. The other big assumption here is that if you do increase the scale by 200-fold, it is economically viable. I mean, if you've got if you only have 20 patients per year, you're not going to need to increase the scale by 200-fold at all, are you? So that's just an assumption of how to how that would contribute. A third, a lower lentiviral vector requirement, so something like CAR-T, the contribution is around 20% of the raw material cost, similar to what we saw from the slides from Cytiva. And if you were to increase that scale from 10 litres to 500 litres, you can reduce the raw material costs of lentivirus from 20% down to 2%. So, as building out the capacity will drive down the costs while increasing your capacity. It's a logical development. Again, it assumes you can work with suspension cell lines, and also the capacity does fit with what you actually need. And for me, that's quite that could then drive back. If you are increasing capacity, it means you can drive down this 23% all the way down to potentially 2%. So, what was it? What was always kind of been curious to me and to proteomic, and what I liked is, within this article the authors did start to look into it, is why have titers been overlooked, or why are they still being overlooked? You've got, if you do want to throw, if you do want to go back from if you do want to increase your scale from 50-fold, you do have to go from a 10-liter up to 500 liters. So you're going to be buying a bioreactor for however many hundreds of thousands. You've got the footprint in your lab. You might need to build a new lab or hire out new space to actually to deal with it. And you're looking at 12 months, 18 months of process development to switch to that. And to me, that seems like quite a lot of effort when you could just increase titers in other ways. And when they looked at this, if you increase viral titers by as little as 30%, I mean, so it's going from 1 to 1.3. It's a minuscule amount. You can potentially, if you're working at a 10-liter scale, very small scale, you can increase the actual output by 66%. If you're doing that on a larger scale, 200 liters, you can increase the output by 42%. So while building out clearly has advantages in terms of you can build capacity, you can reduce the costs, so does increasing titers. And the question that we, as Proteonic, we're asking people is, can we achieve better titers for people? And what would that impact be, both for impact capacity improvements and also cost improvements? So what's the impact of improving titers? So this data here is so you're looking at taking that 23% of lentiviral costs of the manufacturing and taking it down to 1%. Can you reduce that 23% down to 1%? And if you can, what titer gains do you need? If you're working on relatively small scale, so the 10-liter cell factor or hollow fiber fixed bed, you need a 10-fold, you need a 9 or 10-fold titer increase to actually hit to hit enough viral vector particles to get the costs down. If you're working on larger, so anything from 300 liters to 2,000 liters, you need a 3 to 4-fold increase of titers to achieve this and go from 20% cost down to 1% cost. So for me, that's not huge titer gains required to actually reduce the cost significantly. Now, it's very easy to, it's kind of, I don't know, for proteomics, it's easy to kind of go, yes, well, everyone should be focusing on titer improvements. That's the best. That looks good. I mean, you've got, so in titer improvements, you can optimize media, you can optimize transfection processes, or you can optimize the vector. It's relatively fast, 6 to 12 months. There's not a lot of resources required to do that, and there's no to zero capital equipment expenditure required. There's obviously some process changes because you may need to use a new cell line or a different media, and obviously depending on where you are with your program, there may be regulatory considerations to look at. On the bioreactor choice, once you've done it once, it builds it's very expandable in the future. If you've taken 18 months, 2 years to install a 2,000-liter bioreactor, then if you can then double that to 4,000 liters, then 8,000, 16,000 relatively easily, because you're just copying the processes from before. The copying, though, is obviously you need the footprint and building infrastructure needs to be there. The first time you do it, it's going to be a relatively large scale process change, and it will take time. For me, I don't, are they mutually exclusive? You have to go one way or the other way. If you look at if you model this and you look at the titer versus the bioreactor approach, say you go down like, okay, we're going to focus on improving our titers. And it should be relatively fast in terms of seeing an impact. And maybe after 1 year you could see an increase, so that's the relative manufacturing units, from 1 up to a 10-fold increase of titer. After 2 years, maybe there's other technologies, other media optimizations or cell lines you could use that gets you to 15, and maybe there's a gradual increase. It will tail off at some point. It could tail off at 20-fold increase of titer. It could be at 30-fold. It's the thing is that after 5 years there'll be new technology again that will be trying to push titer increases, but there's always a limit. Cell factories tend to have a limit of what titer you can hit. On the other side of things, if you look at the trying to bring in a bioreactor that's 20 times bigger than what you're doing, it may take 1 year, 2 years, or it may take like 3 years to actually install the first one. Once you've just installed the first one, you've got a massive jump. Then after 1 year or 2 years, you can add another one, and you can add another one. And it's quite easy to stagger them and increase capacity that way. Now, if you combine the two of them, so if you have both the titer and the capacity, by year 3, you could potentially have 400-fold increase of titers. I mean, that's 400 increase of manufacturing units, and then that can then increase dramatically year on year. And so, while you can go down the route of expanding capacity through just simply looking at the bioreactors, you should also be looking at, okay, what can we do to the cells? What can we do with the media? What can we do with the transfection aspect? And then what are we doing in terms of the actual viral vector technology we're using? Now, of course, as if we are able to increase the capacity and titers this impressively with the new technologies, you're going to have to then consider, okay, what's going on downstream and what happened in quality control. I mean, I still get surprised when I talk to people in the downstream that you can, if you get 40% recovery, that's sometimes considered good, because it means that you're losing 60% of what you just made is just either not coming off the column or is lost on the column. So clearly there is areas of improvement on the downstream as well. And there's good, again, there's companies out there doing quite very nice work to improve that efficiency. And then the other aspect is quality control and analysis. Again, I was at a conference earlier in the year where they were saying that in their batches, and they were doing quite small batches, but their batch release, they were using up to 20% of their material in quality control to actually check for all the functional testing, the safety testing, titer determination. They were using 20% of their batch to actually then release that viral vector, which means that if you've lost 50% and then you're losing a further 20%, if you've lost 50% in purification and then a further 20%, you're actually then getting 30% of what you started with, which is it's certainly room for improvement. However, having said that, the more you put in at the start, the more you will get out at the end. So there is a balance of where you do focus to get the increase of viral vector for the cell and gene therapy. Now, this is probably one of the biggest challenges that Proteonic faces, and I'm sure others do in the field. It's also if you look at the percentage cost of viral vectors in early development, it's 10%. It's not as significant as say like the labor or the media. I mean, it's kind of like you just see it, but okay, yeah, we are spending money on the production of viral vectors, but it's not a big issue. However, people do need to be aware that as you progress through to late phase clinical development, that cost is going to become much more significant, and delaying it has significant associated risks. And it kind of comes back to what I showed at the beginning, is if there are ways to mitigate the viral vector manufacturing as early as possible, you will avoid having manufacturing shortfalls in the end. I mean, imagine if J&J or BMS had the foresight to start looking at viral vector titers 4 or 5 years ago. A lot of these headlines would not have appeared, because they would have got such heart. They would have had significantly more robust manufacturing pipelines. So this then brings me around to what Proteonic are doing to help to solve this issue. So I mentioned at the start that we are a vector technology company. So we have technology to improve the transcription and translation of any gene in a mammalian system. We've been working with a number of partners. Most recently is with NextGen, also based in Leiden, to look at increasing titers of lentivirus and AAV. And what I'll just do over the next couple of slides, and before I conclude the presentation, is just show a little bit of the technology we have and the data we have generated. So Proteonic, we have what we've called 2G unique elements. It's made up of a transcription enhancer element and a translation element. The transcription enhancer element is essentially a dual promoter, and so you get a doubling of transcription, and it's actually considerably more than doubling because there's a stoichiometric effect of bringing more transcription factors to the site of transcription. It's epigenetically optimized, so if you're people that are developing stable producer cell lines, it will remain in an open chromatin structure and be transcribed as needed. It can be linked to inducible promoters. So again, for stable producer cell lines where you need to be able to switch on and off, it's possible to do that. And then downstream we have RNA elements for improving the messenger RNA into the cytoplasm, and then also working with ribosome affinity sites to increase ribosome coming to messenger RNA, which then also helps with stability. Now these elements are relatively small and can be added to ALV lentiviral or AAV systems. What we have found, working with our partners, is that if we add these elements to the packaging vectors, we do see an increase of titers and a considerable increase of titers. And what's important to note is the increase in titers is also increase of capsids that still have DNA within. They're not empty. And so, while we've done quite a few experiments where we can increase the amount of lentiviral particles with lots of empty capsids, but clearly that's a false economy. So we are balancing very carefully of increasing the titers while also making sure that we keep the quality of the capsids in place, and so they're ready to be used for transfection. So here's example 1 of 2. This is where we've got so we started with our lentiviral control. We added in the 2G unique elements into the promoter of the packaging genes, and we see a 4-fold increase of titers. So if you think back to what talking around the cost reductions, you need a 3- to 4-fold increase of titers to go from 20% cost down to 1%. So with our technology, we are able to achieve that. And this is actually quite early data that we got from about 12 months ago, and since then we're looking at titer improvements of maybe 6-fold. There was one that came out at 10-fold. It depends on the different systems, but or different cell lines we're using and starting. It's always relative, the starting material, but we are very confident of what we're able to achieve, certainly in lentiviral and AAV. For AAV, we're doing similar, where we're looking at improving the titers as well. We also decided to have a look at, can we increase the payload? Because often with AAV gene therapies, if they're delivered on a tissue-specific promoter, those tissue-specific promoters can have low expression and can reduce the efficacy of that therapy to the patient. So instead of looking at increasing the titers, we were looking at increasing the expression of the gene of interest or the payload of the therapy. and whether it was whether the payload was delivered through a CMV promoter or tissue-specific promoter. Again, you can see relative to the starting point, when you add the 2G unique elements, you see a 3-fold here increase and a 2.5-fold increase here. So either approach, we can we can start to really help to drive changes and improvements forward for the gene therapies. This now brings me to my last slide, and so we are 2 years we're 2 years into our journey of moving from bioprocessing and CHO cell lines into cell and gene therapy and viral vector manufacture. We have a early access program that we've opened up to let partners work with us to get access to our 2G unique technology. If you are interested to learn more about the technology, or you think actually this could be quite interesting to slot into your current manufacturing platform, then please email me directly, or you can register via the website. And that now concludes the webinar. If you do have questions, I will be very happy to take them, and I'll see if I can put my video back on so you can see me again. I just have to find the Q&A part. Okay. So there's a question that's come in. Let me just bring that up. So the question is, what are the most significant challenges in scaling up lentiviral production for clinical applications? That's a good question. And so for me, what I would say for that is, let me have a think. So I guess you've got several challenges when you do scale up. I touched on one of them when I was showing the data. Is the first part is probably the vector quality and potency at larger scale is crucial. There's if you are increasing the capacity and then the vector quality is impacted, then you have an issue with your release, your QC and release criteria. The next aspect is probably the complexity of the purification process, because you can end up with a lot more. You are going to have a lot more material to then purify, and you can't use the same size columns. So you're going to have to you have to certainly scale and make sure you have a robust method in place to deal with that. And I guess the third is as you do scale, there's going to be regulatory considerations to think about because you're going to have process changes. You have to go back on that. Hopefully that's answered that question. I'll just check if there's other questions coming in. Okay, there's another one linked to the quality here. So the question is, what quality control measures are essential to ensure the safety and efficacy of lentiviral vectors? I mean, this is a little bit outside of my my expertise area, but I guess I mean, yeah, for me, you've got to, for any QC, you're going to be wanting to determine your titer. You're going to be looking at purity testing, ensure that you've removed any contaminants such as host cell proteins and DNA. You're going to want to make sure that they're functional. So whatever functional assays you have set up for looking at the biological activity of the vector, and then you've got safety testing for replication competent lentivirus and endotoxins, and the last part will be, I guess, linked to sterility testing. So making sure you know what the sterility is of your vectors if you are putting them in storage. How long can you store them for? How long can the batches last? Any more questions? Looks like that may be the last question. So again, thank you very much for joining me today for my webinar. If you do have any questions afterwards you think of, please feel free to email me. If you do have interest in learning more about proteomics, vector technology, I'll be very happy to help. So with that, I will thank you, and hopefully get to meet you guys at a conference sometime or chat later. Cheers.